Okay.
Start now, Prof.
Okay. Bismillah ar-Rahman ar-Rahim. We start today, inshallah, our ICA webinar series number 5, Bioeconomy, Food Security, and Healthy Life. And our guest today is Associate Professor Dr. Mailin Misson. Dr. Misson, she is a senior lecturer in biotechnology research in Institute, University, Malaysia, Sabah. And she holds several research grants as a project leader, and her research related to enzymatic and microbial fermentation technology, bioprocessing, and nano biotechnology. Her research work is focused on conversion of the waste into valuable product. She got her PhD from Adelaide University in Australia in 2016. And she published several book chapters, technical papers, and peer-reviewed in indexed publication, more than 70 publications until now. And she actively involved in the various community service, actively committed service for her research institute in the management position in the postgrad program, and coordinating different programs in the university, and she was the Deputy Director for Research Innovation 2021-2023. Dr. Misson, she have high reputed publications in the field, and she have H index of 17 for a time being. For having a good webinar, we should also having an excellent moderator for today. We have Dr. Zalina Othman. Dr. Zalina have her PhD in bioprocess engineering in the field of recombinant products productions and enzymes productions in E. coli. And her PhD was in collaboration between UTM and Lund University in Sweden. Dr. Zalina, she interested generally involved in the application of bioprocessing strategies to improve productivity and combining the optimization of the production media and process variable towards scaling up studies of the microorganism for food and agriculture industry. Her current research focuses on biorefinery of agro waste for the development of the biological control agents for human health and sustainable agriculture application. She have also highly significant publications in the field, and she currently have H index of 21. And I'm sure today we are going to enjoy all a very nice webinar with the highly important topic nowadays for exploring Sabah'S microalgae as a gateway to sustainable value-added products. Thank you again, Dr. Mailin, for accepting our invitation, and also for the moderator of the day, Dr. Zalina Othman. Please.
Thank you very much for Hisham, our director, ICA Pagoh, and also my supervisor. Thank you for you sharing your experience with me. And also, I would like to say good morning to Dr. Mailin. And also, Assalamualaikum to all of you. How are you, Dr. Mailin?
Good. Thank you so much.
Okay, for your information, Dr. Mailin is our UTM alumni. She is alumni for industrial biology courses at Faculty of Science. I already told my colleague at Bioscience department that today we have our alumni, our students. Okay, today we are proud to have you, Dr. Mailin. So in order not to waste any more time, I would like to invite Dr. Mailin to present her research activity under the title of Exploring Sabah's Microalgae, a gateway to sustainable value-added product. So, Dr. Mailin, the floor is yours now.
Thank you so much, Dr. Zalina, our moderator this morning. And first of all, I would like to say thank you to ICA, especially the director of ICA, for inviting me to be one of the speakers for your webinar series. And it seems like this is probably the first for this year, an opening for the ICA webinar. And I feel honored to be here today. I will share my slide. Maybe Dr. Zalina, during my presentation, should my voice not really clear or the slide isn't moved, just kindly notify me, or I presenting beyond the given time, just let me know. All right, let me share my slide. To record. This computer, hang on. What is it? Okay. Can you see my slide? Dr. Zalina, can you see my slide? I can't hear you, Dr. Zalina. Can you
Oh,
see?
sorry. No,
Yeah.
not yet.
Not yet.
Yeah.
All right. Because just now I received the notification like recording the system. I'm sharing it at the moment. Hang on, but very sorry.
No, it's okay.
Screen. How about now? Can you? How about
Yeah, I
now?
can see, but not your slide.
At the moment, not the slide.
Yeah, not the slide.
Oh. Okay, let me stop sharing first.
Okay.
Right. Entire screen, Microsoft PowerPoint. Actually, I'm presenting my screen. Just now I received notification like you are recording, but my MacBook, MacBook security is very high. We tested it before, it's work.
Yeah.
Yeah, it work.
Maybe Suhail can help us to share with the slide that you have.
Yeah, can I share the slide too, just in case it doesn't work? Just trying to sending to the, just in case I am trying, but hopefully it works for a short while. All right. Cannot see, right? Yeah, this. Okay, I have sent the slide to Dr. Nurliza. Probably the Rosetia can see. Yeah, I have shared to Dr. Nurliza.
Okay, we wait for
Stop
Dr.
sharing.
Lisa for sharing the slide.
I'm very sorry for the technical error, technical problem.
It's okay,
And hi to
you
those
are still
available.
cool.
I would like to say hi to those available online. I could see maybe my former lecturers from Faculty of Science, UTM, or my fellow colleagues from IBD. It's my great pleasure to be here to share our piece of finding here in Sabah. And I hope that through this presentation today, it will open up a room for collaboration between UTM and us here in Sabah, because we have the resources and probably the expertise from there. You can come and work together with us. Yeah, the Universiti will help me. So basically for today, what I would like to share with you is about the wonders of microalgae. Sabah is rich with resources. We have this fresh water, we do have this marine water system, that these are exceptional sources of untapped microalgae resources that probably offering us various applications ranging from renewable energy, nutraceutical, cosmeceutical, what else? Aquaculture feed. There are various of application. Microalgae seems like a very small organism, but it's very powerful in giving us a lot of products. Yeah, so that's what I would like to share today. We have started this work since 2013, if I'm not mistaken. Even started by my colleague, she's the expert in microalgae here in UMS. And we have several students have graduated in this field. And our team is beginning to expand, especially after we received the research grant from Petronas last year. And hopefully that we are able to work with the state government as well, so we can use this very powerful organism to serve our state. Yeah, that's the dream, Dr. Zalina, and hopefully we can make it available in future. Yeah. All right, the slide is there.
Okay, Dr. Mailin, you can start now.
All right, explore. Actually, I have started. I thought I have started. Okay, exploring Sabah's microalgae gateway to sustainable value-added products. All right, that's the title for my presentation today. Okay, next. Okay, I think it won't be any complete presentation without introducing my beautiful university, Universiti Malaysia Sabah, which is located here in Sabah. As you can see the photos here, our university is located nearby the sea. Like our institute is facing the sea. It's very beautiful and with a lot of flora and fauna surrounding it. And this is one of the best place for the sunset view. If you have time to come and visit us, don't forget to visit our beach. This is the best place to see the sunset. All right, next. And I'm attached with Biotechnology Research Institute, one of the institute in UMS. We have 4, which is one of the institute has been upgraded into high COE recently, and we are one of the center of excellence conducting strategic research, capitalizing the rich natural bioresources of Sabah. So one of the resources that we are currently investigating is the microalgae. Right. Okay, next. Okay, our research focus and niche area, including agrobiotechnology, healthcare, agrobiotechnology, healthcare, and industrial biotech. So for my field of research, I'm covering both the agrobiotech and industrial, and a little bit about the healthcare as well. So our niche area, we are very expert in molecular genetics and molecular microbiology. We have conducted various research to provide solutions for the health, agriculture, and environmental, including like taking the blood sample of our ethnic groups in Sabah. You know, in Sabah, Dr. Zalina and the other audience, we have, I couldn't memorize how many ethnics that we have. And that is the one of our experts here have conducted the research, including the proboscis monkey, our commodity like rice, pineapple, microalgae, seaweed, and many more. Yeah. Right, next. These are our research area. So you see, maybe there is a researcher out there, you see which field you are in, and if you would like to collaborate, work with us, we have experts in micropropagations and tissue culture. We also have experts in natural product and drug delivery, molecular genetics, molecular microbiology, and I'm from bioprocess engineering, and also we have expert in biosensor. And okay, our lab, we have 10 labs. Okay, next. It include genomic, microbiology, biochemistry, fermentation, pilot plant. We also have NMR. If you have samples you would like to characterize using NMR, we do have the facilities here. And we are very proud to share with you. We have this facility, BSL3, Biosafety Level 3 lab as well, which we can use to study infectious disease like the most recent COVID-19. And also several support facilities. Right, next. Yeah, this is the BRI COVID team involved in during the COVID pandemic. We were among the I don't know whether maybe the only or among a few public university that involved in analyzing or receiving sample for the COVID testing. Yeah. At first, you know, when the COVID first attacked us back in 2020, we have no idea how dangerous it is, how we can cope with that. And thank God to this team, a very committed days and night, they serve our state especially. Right, okay, next. And we also do, we provide academic program as well. We focus on postgraduate. We don't teach undergrad here. We have Master of Science, Molecular Biology if you want to do coursework, and also research, Master of Science and also Doctor of Philosophy. All right, next. These are our researchers with various expertise, range from molecular genetics up to gene therapy, metabolomic, virology. We have 4 professors, associate professors, and also senior lecturers who actively doing research here as well. Okay. Next. And yeah, it's wonderful to share with you. Every year we were evaluated for MIRA contribution. So BI is among the highest contributor for MIRA for UMS for 2 consecutive years. We have awarded 6 score MIRA tertinggi, 6 stars. Right, next. And also our students, it's very competitive. Dr. Rafida is our very proud student. She won this 3-minute thesis presentation national level last year. The winner, if I'm not mistaken, from UKM and UITM, the 3rd place UITM, second from us, from our institute. Right, next. I think the last slide for introducing our institute. Yeah, they are among our students, which is very excellent, which has been awarded with Anugerah Thesis Terbaik for Sarjana and also PhD, both from our institute. Okay, so all right. To start my presentations about exploring Sabah microalgae gateway to sustainable value-added product, I will start with the introduction, and we'll give you some overview of our existing, our ongoing research project, and the strategies on how we can cultivate microalgae for high value added product based on our research, our own research finding. So I will share with you the strategies and finally the way forward. Okay, next. Okay, you can see the map here, Sabah map. From the map itself, it can tell you that our place is surrounded with various and abundant seawater resources. And if you go in the middle part of Sabah, there are various or many types or various resources of fresh water. Like we have this Kinabatangan River and many other. And this includes like a marine, river, lake, wetland, island, mangrove, or even hot spring. We do have it here. When I climbed the Mount Kinabalu a few years back, even at the top of the mountain, we could see like a pool of water, which I think like if I was able to take and bring some sample back to the lab, maybe we can see or we can get some, like bacteria which has been adapted to a cold environment for so long. So these resources are diverse, a source for diverse microalgae species that can produce various value-added products. All right, next. Okay, introducing to you what is microalgae. Some people think like microalgae is macroalgae. Algae can be divided into two, micro and macro. Macro is like seaweed, rumpai laut. But for micro, it is a single cell or multicellular photosynthesis microorganism. When we cultivate the biomass, we can harvest them and it can produce various value-added products for various applications. Okay, for example, microalgae can produce lipid. Lipid is a valuable product that can be converted into bioethanol or biogas or biodiesel, which can contribute for the renewable energy or bioenergy. Other than that, as you can see the photos next to at the screen, microalgae biomass can produce various types of nutrients, macro or micronutrients or metabolites, which is very beneficial for aquaculture feed. Yeah, other than that, if you can see the photo, the screen here, there is microalgae that can produce pigment, either green color or red color. This pigment can be incorporated in cosmeceutical products. Yeah, so if there are researchers out there which is currently doing cosmeceutical, maybe we can work together as well. We can extract this, we can take this microalgae and incorporate into your product or into as a nutraceutical to enhance the value added of your nutraceutical product. Because microalgae, their product possess omega-3 fatty acid, vitamins, minerals that essential for human health. And yeah, finally, the bioactive compound. It just like if you heard about spirulina, we can pack them in a pill and we can consume that as our daily supplements. Yeah, right. Next. This is the summary on how beneficial, how microalgae can grow. It requires carbon dioxide, it requires light, and under stress condition, it can produce product that we desired for. But for microalgae biomass, the photos here, this is microalgae biomass. It also can be directly used, for example, as human food, or animal feed, or as a food supplement. Or the product inside the microalgae biomass can be extracted. We can extract them out. For example, lipid, that we can convert them and we can use them for biofuel application. And the other important metabolite compound that we can extract are including PUFA or antioxidant, vitamins, anti-cancer drug, antimicrobial drug. That is what we would like to explore at the moment. We explored its application in terms of renewable energy. We are also exploring it in terms of its potential for aquaculture feed and also its application for, we call it like for bioremediation, to treat waste and at the same time to produce product. Okay, next. Yeah, this is our ongoing research. We have been doing research on microalgae for renewable energy applications. This is, I think, more than 10 years. Microalgae for wastewater treatment, we have grown microalgae and POME, palm oil meal effluent. So at the same time, it producing product, it also reduces the BOD and COD content of the palm oil meal effluent. So coming soon, we're gonna grow or cultivate microalgae and sewage for sewage treatment, and perhaps that we will be able to collaborate with the relevant agencies here in Sabah for that purpose. And we do have now a PhD student currently doing research for aquaculture feed. Right, next. Okay, our work, we started from scratch, which is from the sampling. We do sampling in both marine and also freshwater, including coastal, open sea, or estuary. Later, I explain to you what is coastal, what is open sea, and what are the significant when we take sample from coastal and from open sea, what are the different, and freshwater, pond, lake. And after that, we bring the sample to the lab, we do the isolation, we do the culture establishment. Yeah, it's pretty challenging actually, because when we do isolate, we bring the sample from the wild environment and bring to the lab. The challenge is we need to provide the environment here in the lab that mimic to the environment, the other environments so the microalgae can grow. Yeah, and after that, we characterize the morphology, we identify the species. From the species, by knowing the species, we can correlate that, oh, this is beneficial for renewable, or this is beneficial for aquaculture feed. And definitely the growth characteristic, we need to characterize them. Same with bacteria as well. For microalgae, they are fast grower, they are slow grower. For industrial application, definitely we would like to find candidate which is a very fast grower, which is can increase the biomass productivity, or yeah, to enhance the productivity of our desired product. And we did characterizations including lipid, up to the fatty acid profiling actually, protein, carbohydrate, pigment, fatty acid, and carotenoid. Yeah, why pigment is important? Like for animal feed, sometimes when we go to the market, we see the fish. We would like to see we evaluate the fish based on its color. So actually pigment, when we incorporate in the aquaculture feed, so it can, it can give like the interesting interesting or attractive appearance of the fish. It looks like a reddish color if we introduce the red pigment. So this is a very big potential for microalgae actually. Right, next. Just to show to you the sampling area that we have covered before in our west coast area of Sabah, from the northern part up down to Kuala Penyu. And for every places that we went, it's covering coastal area, the open sea, and also estuaries. And some area in the middle here, Tambunan, Keningau as well, collected some samples from kolam ikan, for example, or lake, recreational lake. Yeah, so that's give us a diverse area of collecting sample collection. So it high tendency for us to get a various unique microalgae here in Sabah. All right, next. Okay, for coastal area, coastals, by definition, it is a land meets the sea. Yeah, nearby the beach. When we bring our family for picnic, that's we call it coastal area. If we take sample from that area, that we call it coastal. This area highly influenced by tides or temperature and nutrient inputs. So this can impact the microalgae growth and metabolite production, including lipid. Let's say today we went to take samples during high tide, and in few weeks ahead we go take sample again during low tide. The possibility of getting a different type of microalgae is very high because of the high tides and low tides. The seawater is coming, or maybe microalgae from very far from Peninsular Malaysia can come here, so we can isolate that. Meanwhile, for open sea, which is very far from coastal area, this area actually has been exposed to low nutrient availability. So we can isolate microalgae that has been exposed to that kind of environment, which is probably has been, in their life, they have acclimatized themselves to respond to nutrient stress. For estuary mean river meets the sea. If you go to the sea area, you can see like from the sea, there is a small river going in. So it is characterized by fluctuating salinity and nutrients levels. During low tide, the salinity is different. During high tide, salinity is different. And we experience it by self. When we did sample, we analyze the water sample that we take, and we can see that the salinity level is different. Even the pH of the water resources also different. Meanwhile, for fresh water, like ponds. It is artificial or semi-natural water bodies. Eutrophic condition in pond promotes the growth of microalgae, like lakes, which is freshwater bodies, natural lakes. It can be divided into two: either eutrophic, which is rich with nutrient, or oligotrophic, which is we consider tasik mati, which is there are very minimal amount of microalgae that we amount in that water resources. So the microalgae that we can isolate also will be different. So I hope this can give you some overview on the importance of where or importance of the sources of the sampling area. All right, next. Yeah, okay. Just okay, for the couple of slides after this is just want to show to you how we did that. This is the sampling. We use the plankton net. This is, we call it plankton net, specifically a device that used to isolate microalgae. At the bottom down here, it's like a metal base with specific size that will help us to isolate microalgae. Right, next. And then the sample we brought back to the lab, observed under microscope like this. It's a bit challenging to capture microalgae, especially those motile. You know, we need to use a very small glass like this and capture it and then transfer into well plate. So sometimes, you know, it's very difficult, but that's the challenge. But it's make us like happy if you are managed to transfer them into a new plate. Next. Okay, this is the well plate. You can see there is a various colors, some still yellow, some still look green. That shows actually how the cell the cell density. When it's green, that shows it's really very dense, and you need to redilute and transfer into another well. Dilution is important so that we can get a very pure culture. Right, next. Yeah, this is how pure strands looks like. If we manage to get something like this, then our samples are ready to be identified. Next. Yeah, established in a flask like this. So this also poses challenge when we transfer from small plate into flask. Some of the culture couldn't survive. Yeah, maybe because when we put them in a bigger flask or in a bigger environment for them to grow, they need to, you know, like exposed to temperature or the media itself, or even the shaking. It can put them under stress or can lead to the death of the cell. Next. Yeah, just to show to you the identification of species via 16S RNA. Next. Yeah, mass cultivation in rack chamber. We did something like this. You can see from the color itself, it tells us how dense the microalgae inside it. Right. Okay, next. Culture harvesting, and proceed to the next slide. We see how the microalgae biomass looks like. This is how the final product we get, microalgae biomass. For those applications which can be directly consumed, we can straight away use this. Maybe this biomass can be incorporated into animal aquaculture or animal feed supplement, as a feed supplement, yeah. Or if we would like to extract the bioactive compound from this biomass, we need to have a specific method to do that. Yeah, like for lipid, we need to have like a specific solvent at specific ratio so that we are able to get the lipid, and finally we are able to profile the fatty acid. Right, next. Yeah, the sampling for coastal, we love to find jetty. It will be easier for us to go somewhere right in the middle so that we can get some samples. Right, next. This is from jetty, just showing to you several ways how we can get the sampling. This is open sea. So if very far from the coastal area, we need to seek help from the fishermen, or we hire a numerator to help us. We train them how to get the sample, how to analyze the water quality. Most important, we can get the sample from various resources. This is from OpenSea. And the next from river. Yeah, from the river itself, you see different color, different properties, different pH, different salinity. Right after that, mangrove. Yeah, Sabah, we have a lot of mangrove resources here. Yeah, so these are maybe the favorite place for the microalgae to grow. Right, next. Coastal. From coastal area, if there is no jetty in the nearby area, what we need to do is we just need to be just like what you see in the screen, just like at the photo here. This photo are taken from 2 different locations where a very nice way, but this one, coincidentally, when we arrived to the sampling location, there were a warning weather during that time. This place located in Kota Belud, which is pretty far from Kota Kinabalu. We have been in the location, so we have no choice. We need to just go there and take the sample. And fortunately, we managed to isolate several microalgae from this area as well. Okay, next. Next slide, please. Yeah, this is Cove, another unique area as well. If you go see, check Sabah map, for example, you can see like a cove where the resources here may probably has been trapped. We call it trapped. It has been there. Like for the coastal area, it's exposed to low tide, high tide, and then maybe some of the microalgae flush away from the area when during the tide changes. But maybe for this area, they are right in that area, they grow in that area, not the tendency for being washed away from this area is very low. So it's a unique area as well that we managed to take the sample. Next. Okay, now this is the strategies that I would like to share to you, strategies in how to cultivate microalgae for high value added product. There are 3 strategies as mentioned in the next slide. Selections of microalgae strains, optimization of cultivation condition, and cultivation of low cost. For your information, what I would like to share to you this morning is based on our own research finding, which has been published, and I summarized the strategies into 3, which is, as you can see from the screen, select the microalgae itself. Selection of the best candidate is very important. We need to find microalgae that can produce the desired product accordingly. And also we can optimize the cultivation condition. And finally, we need to find microalgae that able to grow in pome, for example, or can grow in sewage, or can grow in any aquaculture waste, right? We start with the selection of microalgae strain first. Selection of tropical microalgae species for mass production based on lipid and fatty acid. For what we did, we select the strain based on their ability not only to produce lipid, but we also would like to see how's the profile of the lipid. You know, lipid can be divided into saturated fatty acid, unsaturated, or monosaturated, or polysaturated. So those things are very important when we are selecting the best microalgae strain. But first and foremost, when we have the strain, we need to see the growth rate, whether they are fast grower, whether they are slow grower, because from economical point of view, from the industry, they would like to see, they would like to use microalgae which is fast in growth. If very slow, then it's gonna be reduced the productivity. And secondly, they must be adaptable to cultivation condition. Just like the example that I have mentioned to you earlier, when we bring the microalgae from the environment and we bring them to the lab, some of them are dying. They cannot survive. Yeah, because although we have tried our best to provide environment that mimic to the to their original resources, but you know, when we are talking about labs, some of them couldn't survive. And third, they must be efficient in utilizing resources. Like just I mentioned to you, can utilize low-cost media. They can grow in pome. They can grow in any waste that we introduce to them. And most importantly is actually the product that they are producing. What are the nutritional values? What are the secondary metabolites they're producing? What are the bioactive compound? So from there, probably you can isolate 10, 20, 30 isolates of microalgae. Maybe some of them can be applied for renewable energy. Some of them can be applied for aquaculture. Some of them can be applied in nutraceutical, for example. So, all right, let's see the next slides about the details about this paper that we have published earlier. These are the strains. These are the strains that we, this one taken previously from KK area, various type of microalgae species like Anschistro, Chlorella, Chlamydomonas. Yeah, maybe those who are working in microalgae, you are quite familiar with this, the name of this microalgae species, right? Okay, next. Okay, yeah. NFW1 until SW4 are the strands, the code of the strand that we provide, that we give before we manage to identify the strand. And SW1 is actually Cyticerus mulleri. Yeah, the name is Cyticerus mulleri after we identify. But when we isolate, we just give them code. So you can see here, after we quantify the lipid compound, quantify the yield, the yield, and after that, we analyze the fatty acid, the fatty acid profile. You can see here. There are various carbon like C14, C18, C22, but for lipid application, for renewable energy application, we interested on MUFA, M-U-F-A, mono unsaturated fatty acid, which is normally characterized by a higher carbon like C18, which is oleic acid that I bold here, just for an example. So we can see here from these isolates, we found that SW1, which is Cyticerus mulleri, show the highest potential for renewable energy application. I wouldn't say that the rest isolates are not good. Actually, we need to see the potential. Even like C14, C15, this is beneficial for aquaculture feed, actually, lipid. Yeah, it's beneficial for fish if we can incorporate them in fish feed. Right, next. Okay, the second strategy, we need to optimize the cultivation condition. And this is very important. Why? Because although we have a very fast grower microalgae, although they are producing high biomass compound, biomass yield, microalgae is different with like bacteria. Microalgae, when under optimal condition, optimal growth, they're just producing biomass. They producing a high biomass compound, high concentration, but probably not the product that we are desired for. For example, lipid. But knowing this fact, we can manipulate this. We can use this as a strategy to produce the lipid. I hope you can understand. First, maybe we enhance the biomass production first, and after that, we induce them. We put them in a situation or in a condition where we make them under stress. When they are under stress, then they produce the desired product like lipid. So one of the important factor is nutrient supply, which is the most popular is nitrogen. So to produce high lipid compound, we need to induce the cultivation condition under stress limitation, which is nitrogen limitation, or in the absence of nitrogen. Yeah. Secondly is light. Yeah. Light is very important for the microalgae to grow because it's undergoing photosynthetic activities. Under very optimal temperature, they just multiply to produce biomass, but not our required product. So from there, we can manipulate this condition. We put them under stress light, maybe very high. However, we need to ensure that it's not high enough that can cause to photoinhibitory to the microalgae. Photoinhibitory means it can cause the death of the microalgae because the light is too high, the light intensity is too high. And same goes to temperature, carbon supply, pH, and salinity. Right, next. Yeah, this is the typical growth characteristic, just like a bacteria. They have this lag phase, log phase, stationary phase, and finally dead phase. So under vegetative phase, we can harvest them when the biomass at the highest level. And after that, we introduce them under stress condition, as shown in the next slide. Okay, next slide, please. Okay, like this one is we grow them, or in bahasa pasar we can call it like kita gemukkan dia dulu supaya concentration dia banyak, and lepas tu we put them under stress, so we force this biomass to produce the desired product. Yeah, so this slide is prepared by master student. She is doing two-stage cultivation using nitrogen stress. So this is ongoing project actually from one of my master student. This two-stage cultivation, hopefully she could success and can publish this piece of work. Next. Yes. This is the illustrations of what I have mentioned to you earlier. Under nitrogen sufficient, under optimal condition, under favorable condition by the microalgae, they just multiply. The green color is represented as biomass. They just multiply the concentration from 1 into become 2, 2 become 4, 4 become 8, for example. So the biomass increase, but if you can see the yellow dot inside, that is represented as lipid. The amount of lipid doesn't increase, only the biomass increase under favorable condition. However, under stress condition, which is nitrogen insufficient, instead of multiplying the biomass, they increase the productions of lipid, represented by the yellow dots in the green microalgae biomass here. So here the biomass may decrease, the biomass not decrease, maybe either remain, the concentration remain or slightly decrease, but the amount of lipid it producing increasing. So this is one of the strategy. How to do that? That is the role of researcher, how to explore it further. Right, next. Okay. The effects of light intensity and nitrogen concentration to enhance lipid production in the selected 4 tropical. Just now we isolated quite numbers of microalgae, and after that, looking at the potential for lipid production, we just focus on 4, which is published in this article. Right, next. You see here different nitrogen concentration. It will affect on the lipid productivity. For example, next, there is an error. Okay, for example, for unkissed straw, it preferred the highest nitrogen concentration to produce slightly higher lipid productivity. Meanwhile, for chlorella, require around 5 gram per liter nitrate to produce 159 milligram per liter per day lipid. Right? Okay, next. Again, for this one, this 4 isolate, we also profile their fatty acid. Yeah, and for the C18, among these 4, Citoceres molari show the highest oleic acid production. Right, next. Cultivation in low-cost media. Yeah, right next. This one we grow them in pome. Just I mentioned you just now. Right next. Okay. Pome is very contaminated water resources, and then from the color itself, it is dark in color for those which is very contaminated. Microalgae, it require light to grow, but we tested the microalgae to grow in several samples collected from different ponds from mill effluent in Sabah. For the rest of pond, like pond from aerobic pond, from anaerobic treated pond, or from the secondary treated pond, our microalgae could grow very well. But there is one pond which is the fresh, where the processing, all the processing goes into the first pond that is very contaminated. So that is a bit difficult or a bit challenging for this microalgae to grow. But what we did is we co-cultivate with yeast. Why? Because both of them, there is a synergistic interaction. The yeast producing CO2, it utilizing oxygen to grow, but it releasing CO2 through the respiration system, which is beneficial for the microalgae to grow. Microalgae require CO2, and releasing oxygen with them will the process going on and on until the biomass is increased, and both of them synergistically producing the desired product. Okay, I would like to show you the next slide, how contaminated or how, yeah, this is the pome. Without any treatment, we just directly test. We take the sample here and we grow our culture here. Yeah, this one is for the rest of sample which is treated. The rest of sample taken from different pond. It's easy for the microalgae to grow, but for this one, it's pretty challenging. Okay, let's see how high the COD, BOD in the next slide. Yeah, COD is 50,000 milligram per liter. BOD is 68,000. It's very high. Yeah, very. In fact, this pome is actually consisting of nutritious compound for the microalgae to grow. Like they have this nitrogen, they have this carbon, phosphorus. It's become food for them to grow. However, in a very high concentration, it's not become food. It's become toxic to them. So that's how we need to do the strategies, to strategize on the cultivation so that they can grow well. Right, next. Okay, so we grow them under control media as a control of the study, and also we grow them in a pome media as co-culture and also single culture to compare their performance in single and also co-culture. And the SEM image here shows in single culture of yeast, this is how it looks like. In a SEM image of this microalgae, this is how it looks like. And when we co-culture them, we could see a mixture of like a microalgae and also the spore forming oval-like microalgae. Right, next. Okay, I selected a very brief results on the COD removal. We could see that the co-culture poses slightly higher than the single culture, but this one is the preliminary one. After this, we did the optimization using RSM, response surface methodology, and the co-culture COD removal is very high, reaching up to 87%. And in terms of lipid production, we could see that the co-culture as well produce a higher yield compared to the single yeast and microalgae. Right, next. Yeah, this is the comparison study with others. We see that the COD removal reaching up to 87%, when we have optimized the lipid production reaching 15 milligram per liter per day. Yeah, per day. However, like chlorella, the waste that they use is food waste, municipal, shrimp waste, probably not as highly contaminated or highly concentrated like pome. Pome is the one that we tested here. Is very high in COD and BOD. Right, next. Okay, I think done with what we have done. So now I would like to just to share with you that this work is not ending there. We do have a collaboration with the industry, like just recently with Petronas, they're interested to have a biobanking of indigenous microalgae for potential energy. And next, with the agency collaboration, this gonna be at our upcoming next project, cultivation of the microalgae in sewage effluents for simultaneous waste removal and production of valuable product. But maybe sewage effluent is not that really high COD or BOD as compared to POME. So maybe this one is more easier to find the solution. Right, next. And we have this collaboration with UTN. Unfortunately, my slide just now couldn't be shared. Actually, I have updated this slide. I put photos of when my fellow colleagues from UTM came to visit us here in UTM. I have photos of Prof. Hisham as well when he visited me here back in 2019 before the COVID pandemic started. So we worked together, and previously with Prof. Dr. Dr. Aisha Saidina, Prof. Nasa. She was the one who trained me on trained me in doing research and also publication before I joined IBD UTM. And yeah, and then I'm doing some bio-nanomaterial research as well with Dr. Go Patients from MTech. Right, next. As for way forward, this is our dream. We would like to have probably in future, if we can develop like a raceway or open pond that we can do, put somewhere outside of our building, for example, where we do not need to put artificial light. We just let the microalgae to grow by using a natural resources like this sunlight, and then grow them in low-cost media of like media like aquaculture waste, and finally getting isolating the microalgae products to be commercialized in future. Hopefully that is the direction that we would like to see in future. Hopefully we're gonna have like a collaborator from our side that we can put this into a reality. Right, next. Definitely this project cannot be done without our research team. Proud to introduce my team. We three of us, myself from bioprocess engineering background, and Madam Grace, our expert in algal biotech, and also expert in molecular biology. She's the one who actually started this project, and with Prof. N before, and we continue working as a team. With different expertise, we work together, and finally we reach this stage. And we also have Associate Professor Dr. Wilson, our biochemistry and plant biotech expert. And thanks to our master student, PhD student, and RAs, we have one more here. I didn't manage to put in this slide, but I put in my slide just now, actually, Nadia. And we have several students who have graduated with our team doing a project related to microalgae. Right, next. Acknowledgement to our institute, Biotechnology Research Institute, UMS, and all the research grant that has funded our project all this while, FRGS, UMS, internal grant, and also Petronas grant. Thank you so much. And finally, my very last slide. Thank you for bear with me. I hope the sharing today is beneficial to you, and I really hope that it can open up for any future collaboration between UTM and us here in UMS. Thank you. Back to the moderator. Thank you so much.
Okay, thank you, Dr. Mailin, for interesting research activity. You actually have a lot of questions from audience. But there is one question, not question actually. Maybe he say about Sabah is very beautiful. Is it correct, Dr.
Yes,
Mailin?
truly correct. Yes,
We have to go there one fine day. Okay. Okay.
yes.
There is another question. What is the current microalgae product in our market? I mean, maybe in Malaysia, if possible, you can give an example.
Spirulina?
Spirulina.
Yes. That has been long, long way. Yeah, yeah, has
Doctor,
been.
spirulina is freshwater or marine?
Actually, the microalgae, like chlorella, they can both, can be isolated either from marine or freshwater. So, like us, when we do the sampling here, we can collect the similar species from different resources, either microalgae or marine or freshwater. So the one that produced by, what is the distributor, Elkan Gato, spirulina, I'm not really sure actually the origin where they take the sample from. Yeah,
Okay.
but that what I can share to you, that even the marine and freshwater are two different salinity, but high probability to get similar species. Cuma maybe the properties are different because they has been exposed to different kind of environments. Some in high
Yes.
salinity and others in zero salinity of water.
Actually, which one is more easy to handle? Freshwater or marine?
This one I need to ask my RA and my students which one the most easier for them.
Easy to contaminate, like that, or both same?
They have their own challenges.
Oh,
They
okay,
have their
okay.
own challenges. Yeah, I couldn't say that seawater is the best.
Yes.
But for Petronas, they prefer seawater. Why? Because we have resources, very big resources. In future, if they would like to grow in large amount, we can utilize real seawater. We can take seawater to grow instead of
Okay.
using fresh water. If fresh water, we need to use our own resources bin from
Okay.
Jambatan Air, for example. But for seawater, marine water, there has been we have abundant seawater system in the environment. And then according to them, they don't really interested on fresh water because it's easily contaminated lah. Yeah, during our discussion that time. But it seems like they're interested juga lah for fresh water. Doesn't mean that fresh water doesn't have future. Yeah, everything has their own advantage and disadvantages actually. But
That
for
means
fresh
that
water, yes,
means that once we cultivate microalgae from seawater, the base medium still we are using seawater as a base
seawater.
medium.
Seawater.
Okay.
So what we need to do is probably we just need to add a little bit amount of nutrient, carbon, phosphorus, yeah, all those minimal nutrient, or what we call that. We have this Walney BBM. BBM and Walney, they are two types of medium that we currently using. For those from microalgae, from marine, we grow in a specific media, and those from freshwater, a specific media. So advantage if the microalgae can survive in slightly possess salinity properties, we can just take water from daripada laut lah, Kak Inam, and we bring to the lab, and we put some media for them to grow. Yeah, so that's the advantage lah compared to fresh water. But for fresh water, let's say in future we would like to grow them in waste media, for example, pome, or for example in sewage, which is abundantly available in our environment. We want them to grow. So those taken from fresh water are really beneficial for that application.
Okay. The choice of microalgae depends on where we want to use it.
Yes.
Yes.
And
Okay.
what is the application? Yes, what
Yes,
is the
application.
application?
Yes. Okay. Okay, another question, how can a microalgae be integrated into existing wastewater treatment system to enhance efficiency and sustainability? This is what we just discussed just now.
Mm. Oh yeah. For research study, definitely we do it in the lab first.
Mhm.
In a like in a flask, we have the media. No, we use the waste as a media for the microalgae to grow. Yeah, maksudnya kalau from sewage, we take sewage from the sewage effluent, just put a little bit media, for example, and let the microalgae to grow there. And for the real application, the next step is it's difficult for us to bring the waste from the pond to go to the lab. So we
Yes.
must ensure that maybe we can cultivate them in large amount in a raised pond, for example. Then after that we can apply. We can take this probably down in the pond, let them grow, let them cultivate by themselves in the pond. At the same time, we can reduce the COD, BOD, and producing, and we can harvest the biomass. So I'm thinking of like there's a pond. At the end of the pond, there is an inlet and outlet, right? So at the outlet, probably where we can take the sample, we can do the filtration to harvest the product that produced in that particular pond. But
Okay.
of course, this must be collaboration together, not only by us as a researchers, but like engineers, the industries, the academia, and to make these things possible in real application in future.
Yes, doctor, I agree with you. To go further, we have to collaborate with others.
Yes.
Okay. There is not one question, maybe 4 questions from the same audience. There is, what methods did you use for harvesting and drying the microalgae? And he also asked about how much biomass were you able to obtain when using the shade glass? And then another question is, which microalgae from your study can grow fast and how long it takes? And the last question from him, what are your thoughts on the cost of using microalgae for producing bioproduct?
Okay, we answer one by one, lah kan?
Yeah.
Is it at the chat box?
Yeah, in the chat box.
Okay. How methods harvesting? Sekejap ya. Okay. Just now I captured about the fast grower, right? Maybe you can check in our published paper which one is the produce the highest growth rate. Different microalgae has their own growth rate. Some microalgae can grow 7 days, 14 days, or up to 30 days. Then complete the whole life cycle. But some of them, by 7 days, they has reached the stationary phase. I'm sorry, I couldn't really remember what is the exact values of the specific growth rate. But yeah, that's how I can tell you that there are strains within 7 days, it has reached to the stationary phase, 7 hari. But there are strains can reach up to 14 days to reach the stationary phase. Or the amount of the cell is not, even though has reached the stationary phase, for example, the cell density isn't as high as the others. Yeah. And what did you use for harvesting and drying? We just use the conventional method at the moment, for the lipid, for example, lipid extraction. We use, the biomass just now. It is in the liquid form in the shed plus, right? So we did the centrifuge. We centrifuge them to get the pellet, the biomass. And the biomass that we obtain, we further extract using chloroform. I couldn't memorize it. What is the name of the solvent? Chloroform, methanol, and water, if I'm mistaken, 1:1:1 ratio. for the harvesting. And drying, since we do it in a lab scale, we just use oven dry. After the speed vac process.
Okay, doctor. When you are using conventional method for your drying, for you to dry the microalgae, there is no effect to the yield or the activity of your product.
Probably they got some error because this is based on gravimetrically measurement, right? But we try to handle it as the best we can, and every analysis must be done in triplicate. Yeah, even the cell count. Sometimes I kesian juga lah to our RA because they need to, to cell count is using the hematometer
Yes, I
device. They
understand.
need to observe under microscope. Itulah dia kadang-kadang cakap mata berpinar-pinar. So that's the challenge lah, yeah.
Maybe
And then,
next of your research, you can send your sample to ICA. They have a very huge freeze dryer.
oh, yeah, sure.
Yeah.
When we come to the commercialization stage, true. Yeah, when come to the commercialization stage, because we
Yes.
face the challenge when to do the fatty acid profiling. There is a specific requirement. There is a maximum requirements of how much biomass, berapa gram microalgae biomass that you need to send to the sample for the analysis. So when we produce in mass scale, like 5 liter, 10 liter, 20 liter, or more than that, when we do the centrifugation, it's pretty challenging because we don't have the bigger scale here in our institute. So what our students do is buat sikit-sikit lah, they're using the 50 ml Falcon tube.
Don't worry, doctor. You can export to ICA. Okay. What is the maximum CO2 for the
We didn't use CO2. For your information, for what we have tried before, we don't use CO2. It's just naturally there. Because for the co-cultivation, for example, we want the CO2 supply by the yeast.
Okay.
Yeah, remember just now I did the presentation?
Yes.
There is a synergistic gas exchange between the yeast and microalgae. So we didn't supply CO2. We solely dependent on the CO2 produced by the yeast.
Okay. Does it vary based on the species?
Based on well, I think so, yes. But to proven it, it will be better if we test it different species lah. The one that we use is memang has the potential to for bioremediation purpose. The microalgae that we use, when we have a large amount of strains, kita tengok bukan semua yang kita guna untuk wastewater treatment. We look at the potential, which one is, and definitely before we use that, there is a criteria on how we select, or there is a preliminary study before we conducted that. So yeah, before we come up to this level, there is a preliminary study.
Yes, I agree with you because when we have to supply CO2, it increase our cost of production.
True. Yeah, we that's the dream lah kalau boleh kan. We want to do it naturally. The light is coming from the sun, and then the media coming from the seawater or from the organic waste. Without addition, yeah. Because I always think we are from bioprocess field. We always think about when you add something, it will add the cost.
Yes. Okay. Another question, beside UMS, is there any institute or university in Malaysia that run this kind of research?
I think ada.
From your knowledge.
Yeah. Research in microalgae, is that you mean? If research in microalgae, I think yes, definitely yes. Even UTM also have microalgae research. UKM also ada.
Okay. What is the technical and logistical challenging in collecting sampling the microalgae on the research field area?
Technical and logistical challenge. Okay, I would love to share our very fresh experience. when we do sampling, it's not only in coastal area. I show you just now one of the photo where when we take the sample during a very strong wave, you know, a very strong wave where our researcher need to go to the water area to take the sample, and when they put the, you know, what we call that, the plankton net, instead of taking the sample, they take you know, the sediment of the pasir termasuk di dalam kita punya plankton net. If we have jetty, that's advantageous. When we have jetty, we can use the jetty, so we can go slightly in the middle, so the water, the depth of the water is just sufficient enough for us to take the sample. And secondly, when we go to coastal area, not coastal, open sea, which is about 1 hour or 2 hours from the coastal area, by boat, by speedboat, very far, very far. So there was one time when our fishermen come back, the boat, how to say, pecah, the boat, because of the, you know, so there's a tendency of, you know, like kemalangan, because this is water system kan. We never know what happening. Dia punya boat tu pecah. So luckily nothing happened yang very serious. They managed to come back to the coastal area and they're all safe. What we need to do is just to fix the boat lah. So that is, yeah, for collecting sample is definitely challenging because it's involving various resources. And another thing, another challenge, when you go to estuary. Estuary is when coastal meet sea meet the river. So bila kita masuk kawasan estuary, that is the area of apa ni, crocodile. So that is one of the challenge lah. Challenge, not also challenge, but risk when you take the samples. Yeah, but thank God, nothing happened until now. Although we have conducted from the northern part until the southern part of the west coast of Sabah, nothing happened because we do it with a very high precaution, and we plan accordingly. We let the expert do it. We don't go to the very far. For open sea, we let the fishermen, which is always go there every day, can they're familiar with the place, let them go. But what our role is, we train them how to take the sample, how to do the analysis of the analysis of the sample. Or yeah, if any of our colleagues can coincidentally they hire boat go for fishing, kan fishing, they go to open sea, then we can ask them, yeah, to take the sample for us. That's how we survive lah for the sampling.
Yeah, Alhamdulillah, Doctor. Like the words that always come out from my supervisor. Yeah, you still alive. Okay, last question from our audience. Doctor, will there be any possibilities that some or any of these species could be cultured and co-developed with technologies to absorb pollutants in the atmosphere?
Carbon capture?
Yeah, and mitigate climate issue. If yes, then where are we in the timeline?
Frankly
The question in the
speaking,
chat box.
frankly speaking, I haven't go deeper for carbon capture using my microalgae, but based on my reading in literature, there are review papers talking about this microalgae for carbon capture. Yeah, yeah. But
Maybe next.
yeah, but to the question, to the owner of this question, I'm so sorry, I couldn't really address because I haven't done that. But based on my reading, there are strains that have this capability for carbon capture. Yeah, and that's what, you know, Petronas are looking into at the moment, because they would like to produce renewable an energy which is zero carbon emission.
Okay,
Yeah.
don't worry, Doctor. You still young. Maybe your future research.
But yeah, we cannot do all research at the same time. Probably we can just focus. My field of research is more on production of the valorization of the
Yes.
biomass for production of this product. But probably we can work together. You anytime you know me, you can go to our website. You can send me email. Probably we can work together in this particular field of area.
Inshallah. Okay, for that, thank you very much, Dr. Mailin, for the interesting discussions and sharing of your research activity. Okay.
Yeah, thank you.
I think,
Welcome.
okay. On behalf of ICA UTM Campus Pagoh's team, I would like to say thank you for your sharing today. We appreciate your time, and hopefully this knowledge will increase understanding about the value of biorefinery, especially when we are using microalgae to produce value-added product. For that, on behalf of Prof. Hisham, he sent his regard to you because he's have a management meeting, and hopefully there is a collaboration between UMS, especially with you and UTM. Thank you very much, Dr. Maidi.
Thank you so much. I'm really looking forward for future collaboration with UTM in this particular field of area.
Yes.
Yes.
Okay. Okay, don't end your session yet. We should have photo sessions. Okay, Suhail, you are ready for that? Okay, Dr. Liza, 1, 2, 3. Okay, I give back to Dr. Liza. Thank you very much, Suhail. Thank you very much, Liza, and thank you very much, Dr. Mailin.